and Unit 8 Genetics and Molecular Biology. So first in the Unit 5 Metabolism of Carbohydrates and Lipids. In this unit, in this unit uh, it describes this the unit describes the chemical steps through which glucose is degraded into the simpler compounds, namely pyruvate, lactate, ethanol, and it also describes the pathway for the conversion of the pyruvate back to the glucose, conversion of the glucose to pyruvate and also pyruvate to glucose, and that is called gluconeogenesis, and compare the energetic of breakdown and synthesis of the glucose, explain the energetics of degradation of the fatty acids. So all these things are included in this unit. So first, glycolysis. What is glycolysis? Glycolysis is the anaerobic degradation of the, there are six carbons are in the glucose molecule. Glycolysis is the anaerobic degradation of the six carbon glucose molecule to three carbon lactate molecules in the absence of oxygen, molecular oxygen. It is also called homolactic fermentation. A conversion of the glucose into triose phosphate. Glucose of any monosaccharide. Glucose, the, there are six C6, H12O6. Glucose of any other mono, it is a monosaccharide. It is a monosaccharide like fructose, galactose, and cannot be metabolized until it is activated to the phosphate form. In the first step, glucose, when glucose molecule reacts with the ATP, donor of the phosphate, ATP, it can donate the phosphate molecule. The phosphate group comes from the ATP, adenosyl triphosphate. When the glucose molecule reacts with the ATP, it gives glucose 6 phosphate. You can see in the page number 7. When the glucose reacts with the ATP, it gives glucose 6 phosphate and ADP. This reaction is catalyzed by enzyme hexokinase and glucokinase. Two enzymes are there, hexokinase and glucokinase. This reaction is practically irreversible because of the large energy gap. The del zero value of this reaction can be calculated by making use of del zero value of the hydrolysis of ATP and glucose 6 phosphate. Uh, in the second step, it is an isomerization reaction catalyzed by the phosphoglucose isomerizase glucose 6 phosphate. It is irreversible reversible reaction. Glucose 6 phosphate, it converted to the fructose 6 phosphate. Del zero value is 1.6 kilojoule per mole. And it is the, there is a scheme, it is showing here scheme of glycolysis of alcoholic fermentation, showing entry points of various sugars, how the glycogens, it is converted to the galactose 1 phosphate, and finally lactose, how the glucose molecule when treated with the ATP, it gives glucose 6 phosphate, then glucose 6 phosphate reacted with the ATP converted to the ADP, it gives fructose and 6 bis, fructose 6 bis phosphate, and finally it gives the lactate. So this is the it gives in this scheme. Then glucose 6 phosphate gives the fructose 6 phosphate. Glucose 6 phosphate, when treated with the ATP, adenosyl triphosphate, it gives fructose 1,6 bis phosphate. This reaction is practically irreversible in the direction of formation of the fructose 1,6 bis phosphate because of the large energy gap. This reaction is of critical importance in the regulation of the rate of glycolysis. Fructose 1,6 bis phosphate. When fructose 6 phosphate react with the ATP molecule, it gives fructose 1,6 bis phosphate. Fructose 1,6 bis phosphate in the equation 5.4, fructose 1,6 bis phosphate gets clipped. It clipped into one molecule of each glyceraldehyde 3 phosphate, G3P, glyceraldehyde 3 phosphate, and hydroxyacetone phosphate, DHAP, hydroxyacetone phosphate. The enzyme the enzyme catalyzing this reaction is called fructose 1 6 bis phosphate aldose and simply aldose. So glyceraldehyde and dihydroxyacetone DHAP are frequently referred to as 3 carbon sugars or trioses. These phosphorylated derivative G3P and DHAP are interconvertible. So deoxyacetone phosphate and glyceraldehyde 3 phosphate are interconvertible interconvertible, just like the other aldo keto interconversion like. So this reaction is catalyzed by triosepho, and triosephosphate isomerase. So the conversion of the G3 glucose 3 phosphate into the pyruvate. How the glucose 3 phosphate glyceraldehyde 3 phosphate is converted to NADH. NADH, the glucose 3 glyceraldehyde 3 phosphate formed by cleave of the 6 bis phosphate reacts with the NAD plus and phosphate ion to give 1,3 bis phosphoglycerate and NADH. GGPDH has highly reactive cysteine SH group at the active site. It is a enzyme. It has the SH group in the active site. It has been shown that the reaction proceeds in the daily distinguishable step, oxidation reduction step. Aldehyde group G3P is oxidized. Glyceraldehyde 3 phosphate oxidized to the acyl moiety, which remains covalently attached to the enzyme active site SH group in the form of thiol esters with a concomitant reduction of the NAD plus to NADH. The highly reactive site of the SH group, the enzyme, it is the SH group is there. SH group of the GPDH reacts with the several reagent, iodoacetate, iodoacetamide. When SH group, thiol group, when treated with the iodoacetate, it gives the iodoacid, thioiodoacetate, H plus plus iodine minus. Blocking of this site of the SH group leads to loss of catalytic. Jadi, if we do not use the enzyme, it will lose the catalytic activity of the GPDH. It is well known that iodoacetate and isoacetamide inhibit glycolytic breakdown of the glucose. 1,3 bis phosphoglycerate reacts with the ADP, adenosine ADP in the next step of glycolysis. When 1,3 bis phosphoglycerate treated with the ADP, 3 phosphoglycerate and ATP is formed. This reaction catalyzed by phosphoglycerate kinase and yields 3 phosphoglycerate. This reaction energy conserved in the preceding step is harvested and utilized for a net synthesis of ATP. This is the first ATP generating step of the glycolysis. In the next reaction catalyzed by the phosphoglyceromutase, 3 phosphoglycerate is isomerized to the 2 phosphoglycerate. This reaction requires the presence of enzyme bound to the 2,3 bis phosphoglycerate. A histidine residue of the enzyme active site functions successively as an acceptor and donor of the phosphate group. 2 phosphoglycerate undergoes dehydration to give rise to the phosphoenol pyruvate PEP. The enzyme catalyzing this reaction is called the enolase. Phosphate ester of the enolic compound are also energy rich compound. They have high phosphate group transfer potential. It is made use of the next step to transfer the phosphate ground to ADP, giving rise to ATP and pyruvate. This reaction catalyzed by pyruvate kinase (PK). Pyruvate kinase is the second important regulatory point to control the rate of glycolysis. Metabolism of pyruvate depends on the nature of the living organism and its physiological state. On the rate of oxygen supply under aerobic condition, pyruvate is converted into the acetyl coenzyme and then oxidized to the carbon dioxide by Krebs tricarboxylic acid cycle. Krebs cycle is called citric acid cycle. In the animal muscles, pyruvate is used for this purpose. NADH formed in the oxidation phosphorylation of the glyceraldehyde 3-phosphate reacts with the pyruvate in the presence of the lactate dehydrogenate to form the lactate and NAD plus. Pyruvate when treated with the NADH, it will give the lactate and NAD plus. The resulting NAD plus can be used again for this reaction. Note that none of the reactions of the glycolysis and the reaction given above require oxygen. This permits continuous generation of the ATP when under conditions of limited oxygen supply. Glycolysis is the major source of the ATP during the strenuous muscular activity. In such condition, glycolysis results in the accumulation of the lactate in the muscles. The pyruvate when treated with the H plus, it gives the acetaldehyde. Acetaldehyde when treated with the NADH and H plus, it gives ethanol and NADA plus. The oxidized coenzyme so produce NAD plus can be used again for the reaction given in reaction. This process is called alcoholic fermentation. Another metabolic reaction of the pyruvate is in carboxylation to oxaloacetate by pyruvate carboxylase, a biotin-dependent enzyme. This reaction is important for replenishing the TCA cycle, Krebs cycle, intermediates for providing substrates of the glycogenesis for the glycogenesis. Hmm. Gluconeogenesis. What is gluconeogenesis? Occurs primarily in the liver. Glucolysis and glycogenesis make up a pseudocycle or a pair of oppositely directed reaction sequences. Conversion of a non-carbohydrate precursor like lactate into glucose is called gluconeogenesis. Gluconoglycerol conversion of the non-carbohydrate precursor like lactate into glucose is called gluconeogenesis. Glycerol and some amino acids are other starting material for the gluconeogenesis, which became important under condition of the starvation. Under these conditions, glucose level in the blood have to be maintained at about 80 to 140 milligram per 100 ml by converting non-carbohydrate material into glucose because brain is highly dependent on glucose as the primary fuel. The pathway of gluconeogenesis and entry point of the lactate, amino acid and glycerol shown in the figure 5.2. Okay. How the lactate is converted to the pyruvate and alanine is converted to the pyruvate. When the pyruvate treated with the ADP, pyruvate is carboxylated in the presence of the ATP and biotin dependent pyruvate carboxylate to form the oxaloacetate. The latter then decarboxylated and phosphorylated by its reaction with another high energy phosphate compounds, namely mm, guanosine triphosphate GTP. This reaction catalyzed by phosphoenol pyruvate carboxy kinase. Pyruvate when treated with the carbonic acid with plus ATP, it gives oxaloacetate. How the pyruvate is converted to oxaloacetate? Oxaloacetate it gives the phosphoenol pyruvate, then it gives the 2P glycerate, then finally how it is converted to the glucose, it is given in this scheme. What is Krebs cycle? Krebs cycle metabolize acetyl moiety of the acetyl coenzyme generated from the pyruvate, and the degradation of the fatty acid is oxidized to carbon dioxide and water by a cyclic set of reaction is known as citric acid cycle or Krebs tricarboxylic acid TCA. This is called Krebs cycle. Citric acid, you can call it as the Krebs cycle or citric acid cycle or Krebs tricarboxylic acid cycle. After the name of the scientist who discovered it, this cycle occupies a central position in the metabolic pathways because its precursor pyruvate and acetyl coenzyme may be derived from the carbohydrates, fatty acids, and some amino acids. It is explained above that the TCA cycle, pyruvate must first be converted into the acetyl coenzyme. For this, pyruvate is transported to the mitochondria where it undergoes oxidative decarboxylation and condensation with the coenzyme. Acetate, uh, acetate when uh, treated with the NAD plus coenzyme SH, it gives the CH3COS CoA plus carbon dioxide plus NADH. This reaction is catalyzed by an assembly of enzyme or a multi-enzyme complex called pyruvate dehydrogenase complex. This complex consists of three distinct enzymes held together by strong non-covalent interactions. The three constituent enzymes and their respective prosthetic groups are listed. The complex of the enzyme E2 is present in the center flanked by E1 and E3. Constituent enzyme of the pyruvate dehydrogenase complex. Other reaction of the TCA cycle, it starts the citrate is isomerized to the isocitrate in the presence of the enzyme aconites. The latter is iron sulfur protein in which iron is not complex to any heme moiety. Therefore, it is also referred to as the non-heme iron protein. It has been suggested that citrate undergoes a dehydration followed by a rehydration step with the cis aconitate as an intermediate as shown in the equation 5.16. When citrate treated with the water it gives cis aconitate, again it gave isocitrate. The reaction of the tricarboxylic acid cycle, it gives a how the acetyl coenzyme treated with the NADH, it gives the citrate, then isocitrate, then it's converted when it treated to the NAD plus, it is converted to the alpha ketoglucarate. Then when it again treated with the NAD plus, it is give succinyl CoA, and then it is give fumarate and malate. The cycle is given here. Isocitrate reacts with the NAD plus in the presence of isocitrate dehydrogenase, giving rise to alpha ketoglucarate glutarate, a 5 carbon compounds. This is achieved in two steps. The first step, the secondary alcohol is oxidized to ketone. Secondary alcohol oxidized to ketone, while the second step it involves beta D carboxylation. The next reaction takes place in the presence of another multi-enzyme complex, namely alpha ketoglutarate dehydrogenase complex. This complex is very similar to pyruvate dehydrogenase complex. So, in nature, catalyzed by the reactions as when the alpha ketoglutarate treated with the NAD plus and coenzyme SH, it gives the succinyl CoA NADH plus carbon dioxide. Uh, Succinate undergoes successively an oxidation, dehydration, and another oxidation reaction. In the first reaction catalyzed by succinate dehydrogenase, two hydrogen atoms are transferred from succinate to the flavin adenine FAD to form reduced coenzyme and an unsaturated acid fumarate. It may succinate treated with the it gives the fumarate, then fumarate it gives malate, then oxaloacetate it gives. TCA cycle also provides precursor for the biosynthesis of several other compounds that it performs as anaerobic function. Therefore, it is often referred to as the amphibolic pathway. Let us study how this cycle is able to accomplish this. The formation of the pyruvate from the carbohydrate and amino acid and the acetyl coenzyme from the pyruvate and fatty acids. The acetyl coenzyme is oxidized by the TCA cycle or Krebs cycle. Several amino acids related to glutamates, aspartate, aspartate are metabolized by their conversion into alpha ketoglucarate and oxaloacetate respectively, and both of these are intermediate of the this cycle. The major source of pyruvate is the animal body is the glycotic breakdown of the carbohydrate. Pyruvate is critical for the TCS efficient running of the TCA cycle. Then the study in the fats are degraded to the acetyl coenzyme and later is normally metabolized by the TCA cycle. Carbohydrate indirectly help in the catabolism or burning of the fats in the body cell. This explains the necessity of including a certain amount of carbohydrate in the diets of person who are trying to lose weight by burning some of the body fats. It has been estimated that about one-fifth of the calorie or energy requirement of such person must be provided as a dietary carbohydrate. This ensures the adequate supply of the pyruvate, which helps maintain optimum level of the TCA cycle intermediates. The electron transfer chain, the reduced coenzyme NADH and FADH to produced during the metabolic process of Krebs cycle are subsequently reoxidized by a series of reaction is called electron transport chain ETC, in which molecular oxygen is the ultimate electron acceptor. The pathway is called oxidative phosphorylation. Oxidative phosphorylation, NAD plus is reduced to NADH by a dehydrogenase which removes two hydrogen atoms from its substrate. NADH carries a proton and hydride ion and transfer this to the enzyme complex NADH hydrogenase. This complex has a cofactor that is flavin mononucleotide which gets converted to the FMNS2 by accepting the two hydrogen atom. The hydrogen atoms are next transferred to ubiquinone known as the coenzyme Q. The remaining members of the chain are cytochromes. Electrons are passed down the chain to the cytochromes. Cytochrome is the only cytochrome that can react directly with the molecular oxygen. Rise in the blood glucose level such as following meals is brought down by promoting its uptake by cells by utilization for energy through glycolytic DC and oxidative phosphorylation pathway. Storage as glycogen in the liver and muscles are short-term energy reserve and conversion to the triacyl glycerols in the adipose tissue. At the same time, pathway which generate glucose, such as the gluconeogenesis and glycogenesis, are inhibited. The interorgan coordination of carbohydrate metabolism is mediated by hormones and fall broadly into two groups based on their roles. When the blood glucose concentration is high, insulin secreted by the pancreatic beta cell promotes glucose uptake by cells and its oxidation through glycolysis to the lower glucose level. Gly- Glucagon promotes uptake of amino acid from the blood by liver hepatocyte and converts them to glucose. A rise in the blood glucose level inhibits the glucagon by a negative feedback mechanism. Growth hormone and ACTH, adrenocorticotropin, secreted by the anterior pituitary hormone, rises the blood glucose level and antagonize the effect of insulin as their secretion is stimulated by the hypoglycemia. Thyroxine, a thyroid hormone, accelerates hepatic glycogenesis with consequent rise in the blood glucose. Thyroid hormones may also increase the rate of absorption of the hexoses from the intestine and hepatic glucose beta 6 phosphatase activity. Degradation of the fatty acids. You know that these are the fatty acids are esters of the glycerols and the triglycerol alcohol. This reaction, the triglycerol alcohol, when treated with the when it oxidized when the water catalyzed by reaction takes place when reacts with water, it gives glycerol and fatty acids. Glycerol is phosphorylated and oxidized to the dihydroxyacetone phosphate, which is isomerized to the glyceraldehyde 3 phosphate. The glycerol it gives glycerol phosphate, then it dihydroxyacetone phosphate, and it is converted to the glyceraldehyde 3 phosphates. Conversion of the fatty acid to acetyl coenzyme 3 energy stored in the phosphate linkage of ATP is utilized to drive the formation of the thiol ester linkage between the carboxylic group of the fatty acid and SS thiol group of the coenzyme A. This reaction catalyzed by acyl coenzyme synthetase, also called the thiokinase. Thiokinase acetate, when treated with the ATP, it gives acyl adenylate, and in this reaction, two high energy phosphate linkages have been broken and only one high energy thiol ester linkage is established. Extra energy is utilized to drive the reaction to complete the desired direction. Acyl coenzyme undergoes a sequence for our reaction, namely the oxidation, hydration, another oxidation, and the thiolytic cleavage. It loses two carbon fragment in the form of acetyl coenzyme. The first oxidation reaction is catalyzed by acyl coenzyme dehydrogenase with the FAD as the hydrogen acceptor. The resulting trans 2,3 unsaturated enol CoA adds a molecule of water in the presence of enol coenzyme hydratase. This addition of water is stereospecific, giving rise to L3-hydroxyacyl coenzyme, which is oxidized in the presence of the L3-hydroxyacyl coenzyme hydrogenase with the NAD plus as the coenzyme. The This is the summary of the reaction of the beta oxidation pathway of the fatty acid. How the fatty acid coenzyme converted in oxidation give the trans-enoyl coenzyme, and dehydration it gives the hydroxyacyl coenzyme, then ketoacyl coenzyme, and finally acetyl coenzyme. Mm. So in this, the oils and fats are hydrolyzed by various lipases to give the glycerol and free fatty acids. Glycerol is metabolized by glycolysis. Each fatty acid is converted into the corresponding acyl coenzyme A and transported from cytoplasm to the mitochondrial matrix where it is degraded. Acyl coenzyme undergoes a sequence of four reactions, namely an oxidation, hydration, another oxidation, and a thiolytic cleavage. The oxidation of fatty acid yields large amount of the ATP. In this, so this unit covers the catabolism of the glucose and pyruvates, anaerobic alcoholic fermentation of the glucose, the TCA Krebs cycle or a TCA cycle. TCA cycle occupy the central place in the metabolism process because it helps to oxidize the metabolites formed from the carbohydrate, amino acids, and fats. In addition, some metabolites of cycle serves as precursor in the synthesis of the porphyrins and some amino acids. So, coming to the next unit is metabolism and proteins and nucleic acids. The protein metabolism is essential. The catabolism of amino acid. During catabolism, the fate of alpha amino group nitrogen is different from remaining carbon skeleton. Catabolism also amino acid takes place in three stages as given below. Catabolism of amino acid takes place in the three stages. The three stages are removal of the amino group, leaving the carbon skeleton of the amino acids, breakdown of the carbon skeleton to an intermediate of the glycolytic pathway or citric acid cycle to acetyl coenzyme, then oxidation of these intermediate to carbon dioxide and water in the production of the ATP. Removal of the amino group from the amino acid. The amino group extracted from the amino acid involve the three steps: the conversion of the amino group from the whole amino acid into a single product, glutamate by transamination, the conversion of the glutamate into alpha ketoglutarate by oxidative deamination, releasing NH4 plus, then conversion of the NH4 plus into the non-toxic compound urea, which is the blood carries to the kidney for excretion. The removal of a amino group nitrogen usually occurs by two mechanisms: transamination and deamination, as mentioned above too. What is transamination? Transamination means it is a chemical reaction in which the amino group from an amino acid is transferred to the keto acid. It is catalyzed by an enzyme called transaminase or transferase. Mm, that amino acid to be catalyzed is converted to keto acid. Another keto acid takes place the amino group and form of amino acid. This reaction is catabolized by transaminase. Pyridoxine is used as the coenzyme and the carrier of amino group. It participates in the reaction as pyridoxal phosphate. Deamination. The deamination, the amino group is directly removed from the amino acid. It occurs in the two ways oxidative deamination and non-oxidative deamination. Oxidative deamination catalyzed by two enzyme glutamate dehydrogenase and L-amino acid oxidase. Glutamate dehydrogenase is the more actively used enzyme in the breakdown of the amino acid of the energy. The final acceptor of the alpha amino group is alpha ketoglutarate, forming the glutamate. Glutamate can also undergo oxidative deamination, in which it loses its amino group as the NH4 plus ion and oxidizes back to the alpha keto glutarate. Non-oxidative deamination is the mechanism of direct removal of the amino group from the simple hydrolytic cleavage without any cleavage. Oxidative deamination reaction occur primarily in the liver mitochondria. Most of the ammonia NH4 plus ion formed by oxidative deamination of the glutamate is converted to urea and excreted in the urine using a series of reactions known as the urea cycle. What is urea cycle? The ammonia formed during amino acid catabolism is toxic and cannot be excreted from the body. It gets converted to urea, which is less toxic and easily excreted. The process of conversion of the ammonia to urea. The process of conversion when ammonia is converted to urea, it is called urea cycle or Krebs Hanselate cycle, which is carried out in the liver. This cycle takes place in both mitochondria and cytoplasm. These reactions of the urea cycle is shown in this figure. First step one, formation of the carbamyl phosphate, condensation of the ammonia ion, condensation of the ammonia ion with the bicarbonate ion results in the formation of the carbamyl phosphate with the help of the enzyme carbamyl phosphate synthase 1, which is present in the liver mitochondria. The reaction requires two ATP molecules. So how carbamyl phosphate synthesis 1 carbamyl phosphate mm, citrulline, citrulline AMP implemented, then how it is converted to the urea, it is given here. Uh, in the step to formation of the citrulline, step 2 synthesis of the arginine or succinate, step 3 formation of the arginine, in the step 4 formation of the urea. Enzyme arginase lyses arginine into the ornithine and urea. So this is the step by step it is given below. Oxidation of the carbon skeleton, then degradation of the nucleic acid. Nucleic acid turnover is an ongoing metabolic process in most cells. The nucleotide are of the two types on the basis of the two types of the bases, purine and pyrimidine bases. The nucleotides are generated either by novosynthesis or by nucleic acid turnover and restoring reactions. Dietary nucleic acids survive the low pH in the stomach. These are degraded mainly in the intestine and pancreatic nucleus and intestine. These enzymes help in breaking phosphodiester bonds linking nucleotide. These are therefore hydrolyzed to the nucleosides by a group of specific nucleotidases and non-specific phosphatases. Nucleotide, when react with the water, it gives a nucleoside plus Pi. Nucleosides may be directly absorbed by the intestine mucosa or further degraded to free bases and ribose or ribose 1 phosphate through the action of the nucleosidase and nucleoside phosphorase respectively, as shown in the reaction given. Nucleoside, when treated with the water in the presence of the nucleosidase, it gives base plus ribose. Nucleoside plus Pi nucleoside as it gives base plus ribose 1 phosphate. Although purine and pyrimidine nucleotides are released from the tissue's turnover. The nucleotides which are not salvaged are catabolized and excreted. Both pathways share common reaction involving dephosphorylation, deamination, and glycosidic bond cleavage. Degradation of the purine nucleotide. It is mentioned above that both purine and pyrimidine nucleotide have similar pathway of degradation. Purine and pyrimidine, two bases, and their degradation pathways are same. The purine and pyrimidine bases during degradation converge at the xanthine and uracil thymine respectively. The former are further degraded to uric acid and other end products. Adenosine and deoxyadenosine are not degraded by the mammalian purine nucleoside phosphorylase. These are deaminated by adenosine. It is the catabolism of purine nucleotide. It is given in this scheme. Uh, these are the deaminated by adenosine deaminase and their corresponding anosine derivative for further degradation. Similarly, xanthosine and guanosine are substrate of PNP yielding guanine and xanthine respectively. Guanine is further deaminated to the xanthine by guanine deaminase. Xanthine, like the hypoxanthine, is oxidized by the xanthine oxidase to uric acid. In the man, uric acid is excreted. Uric acid is formed primarily in the liver and excreted by the kidney into the urine. The how this hypoxanthine is converted to the urate, it is given below. It is first converted to the uric acid. It's keto form of the hypoxanthine. Then it converted to the urate. Ketoenol tautomerism happens here. Uh, Xanthine oxidase XO is a homodimer of homodimer uh, it is the fate of uric acid, the purine nucleoside degradation of purine. The fate of uric acid varies among the different group of animal. Uric acid is a purine which is sparingly soluble in water. It is a weak acid that exists in the protonated form at pH below 5.4 and monosodium urate salt and neutral pH. The salt more soluble than the acids. Purine nucleotide cycle, the deamination of the adenylate AMP to anosinate followed by resynthesis of AMP constitute the purine nucleotide cycle. The degradation of the three major pyrimidine nucleotide, as we know, the cytosine is present in both DNA and RNA, where the other two are largely restricted to either DNA, thiamine, or RNA. The ribonucleotide also serve as a variety of other roles, and they are degraded by the same route. Purine and pyrimidine nucleotide degradation, a comparison. There are difference between the two process. The purine bases are oxidized by xanthine oxidase to uric acid, while pyrimidine bases are reduced to dihydrouracil and dihydrothiamine. The reduced pyrimidine ring is split and further degraded to amino acid, whereas the purine ring may or may not be split. The primates, birds, reptiles, and insects excrete an intact purine uric acid. A deficiency of molybdenum affects the purine catabolism, while pyrimidine catabolism is unaffected. Uric acid, the end product of the purine catabolism in some organisms, is also an antioxidant. None of the product of pyrimidine catabolism are antioxidants. So this is the difference between the purine and pyrimidine nucleotide degradation. And interrelationship among metabolic pathway. The interrelationship among the metabolic pathway is essential to interconvert biochemical metabolites as per the cellular need, meet the body's fuel demand continuously, regulates the levels of intermediary metabolites and maintain their equilibrium, coordinate with the various cell tissue and organs for existence, impact normal biochemical environment, and maintain the health. In summary of the relationship among the three metabolism is given in the figure. It is clear that metabolic pathway studies so far it is learned that all the organism utilize the three major foodstuff to form acetyl coenzyme or the metabolites for the Krebs cycle, supply energy upon subsequent oxidation by the cycle. Acetyl coenzyme is the primary link and the degradation of the product of the glucose, glycerol, fatty acids, and amino acids. It is also the starting material for the biosynthetic of the fatty acid, cholesterol, and ketone bodies. You may also observe that one irreversible reaction is the conversion of the pyruvic acid to the acetyl coenzyme. Acetyl coenzyme molecule derived from the fatty acid degradation cannot be directly transferred into the pyruvic acid. Therefore, there is no direct route for the synthesis of the carbohydrate from fatty acid. Plants and some microorganisms that can convert the fatty acids to carbohydrate and proteins utilizes a series of enzymatic reactions not found in the animal tissue referred to as the glycolate cycle. The interrelationship among the pathway can very well be understood and appreciated by the comparison of the food intake by individual in the three condition: overfed condition when it is beyond requirement. In this condition, a limited amount of excess of food gets stored by glycogen and remaining gets stored as fat. Fasting condition is not eating for a short duration. In this condition, the stored glycogen and fat is utilized for energy requirement. Starvation, not eating for a long time. When there is a state of no consumption of food, all the stored glycogen is used. In this condition, the body protein is broken to amino acids that are used to synthesis glucose. The glucose serves as the source of energy for brain and the nervous system. In the liver, fats are converted to ketone bodies, which again are the energy source of the brain. So, mass uh, so these are the comparison of the food intake by an individual in the three condition: overfed condition, fasting condition, and starvation condition. So and the deamination. So in this summary, we learned that protein metabolism, all type of the metabolic pathways are into how the all types of the metabolic pathways are interrelated to one another by several common intermediates, and the dietary nucleic acids are degraded, how the dietary nucleic acids are degraded mainly in the intestine by pancreatic nucleosides. Nucleosides may be directly absorbed nucleosides may be directly absorbed by intestine mucosa or further degraded to the free bases and ribose or ribose 1 phosphate through the action of the nucleosides. And how the purine pyrimidine nucleotides released from tissue turnover, which are not salvaged and catabolized and excreted. During the catabolism, purine bases are oxidized by the xanthine oxidase to uric acid, while pyrimidine bases are reduced to the dihydrouracil. Uh, the fate of uric acid varies among different groups of animals. It is excreted as such primates, birds, reptiles, and insects. In all other organisms, uric acid further metabolized prior to excretion. So units include all these things. Also the deamination of the adenylate and anosinate IMP followed by resynthesis of the AMP constitute the purine nucleotide cycle. And the next unit is the immune system. How the immune system describe the level of the defense present in the body, describe the differentiate the innate and the acquired type of immunity, list of type of cells organs of the immune system, elaborate the origin of the immune system and process of its operation, describe cellular immunity and humoral immunity and the role of T and B lymphocytes, define and describe structure of antigen and antibody, explain process of antigen antibody interactions. So what is immune system? Immunity and level of defense. The immune, all all of us familiar with the word immunity, which is used commonly for an individual's resistance to any infection. The word immunity comes from a Latin word immunis, meaning free. Immunis meaning free or exempt. We often experience that one person may have severe cold and cough while the other person never suffers from cold, although both have the same chances for exposure. Although both have the same exposure, but one is getting cold and other person is not getting the cold. This is because of the resistance toward the infection in one person is better than that in the other. And we say that the person has a better immunity as compared to the other. The immunity can be defined as the ability of the organism to resist a particular infection or toxin by the action of the immune system. The study of the immune system provides the foundation for the very interesting object is called immunology. Immunology, uh, Louis Pasteur is also developed the overall principle of vaccination and contributed to the foundation of the field of immunology. Louis Pasteur. The modern definition of immunity include those physiological mechanisms that enable the animal to recognize the material of foreign to itself and to neutralize or eliminate or metabolize these with or without injury to its own tissue. Immunity may also be regarded as the result of the fight of two opposite forces. Immunity is the fight of two opposite forces, the infected organism on one hand and the host on the other hand. The infected person and who is infecting, that is the host molecule, that the host of the other hand. The result may be complete immunity, partial immunity, or total absence of immunity. Most of us fall in the category of partial immunity. Organisms causing the disease are termed pathogens, and the process by which they induce illness in the host is called pathogenesis. Human pathogens can be grouped into four major categories on their characteristics: viruses, fungi, parasites, and bacteria. A list of these pathogen and diseases caused by them is given them. Pathogens, viruses, varicella, poliovirus, disease, poliomyelitis, coronavirus, COVID-19, human immunodeficiency, HIV, AIDS, rubeola virus, measles. In the fungi, candidiasis, Candida albicans, candida disease, Tinea corporis, ringworm. If these are the disease, these are the pathogens. These are the viruses. Protozoa, Plasmodium species, malaria, Entomobea, histolytica, amoebic colitis, bacteria, Mycobacterium, tuberculosis, TB, Vibrio, cholera. So our body has certain mechanism to counter the attack of these pathogens. That is called defense mechanism. Let us understand these mechanisms. Level of immune defense. There are three lines of defense or the barriers in the body that must be overcome by pathogen before it can cause an infection disease. These are explained in the following paragraphs. Mechanical barrier. These are the first line of defense barrier. Our body is surrounded by both pathogenic and non-pathogenic organisms which through various portals like water, milk, food, air, vector try to enter the body but are stopped by many efficient mechanical barrier which would restrict the entry of such microbes. These mechanical barriers are intact skin, Although unbroken or intact skin is a good mechanical barrier and does not allow any organisms to enter the tissues, sometimes the organisms gain their entry through their hair follicles or opening of sweat gland or through the skin if it is injured or there is any cut or wound. The conjunctive, the conjunctive are the thin membrane that cover the inner surface of the eyelid and the white part of the eyeball. These are protected by motion of the eyelids and the constant washing of the eyeballs with the shedding tears secretions till the organisms may enter through the conjunctive. Mucous membranes and mucous secretion. The mucous secretions of the respiratory, digestive, and urinogenital organs form a protective covering to the mucous membrane. Secretion from the sweat glands, gastric juice, and vagina hold many microorganisms and make them reduce their infectivity. The mucous membranes of the genitalia are protected from upon most bacteria, and mechanical arrangement of the organ structure. Turbinate bone in the nose also form a line of defense. The bacteria, when they enter through the nose, they either stick to the mucous surface of the nose or they take a longer time to reach the alveoli because of its structure. By the time they reach the alveoli, most of the organisms die and very few succeed in entering the organ. Other barriers: coughing, sneezing, shedding, tears, perspiring, salivating, etc., also help mechanically to remove the pathogenic organisms from the entry point. It may be mentioned here that lysozyme and enzyme present in most secretions of the body act as a chemical barrier. Phagocytes, phagocytes being the second line of the defense, these also form a part of innate immune system that you will learn later. If the microorganisms succeed in the passing the first line of defense and enter the deeper tissue, then they are attacked by the specific cell of the body called phagocytes or natural cells, natural killer cells, which include cells reticuloendothelial system (RES). These phagocytes ingest the microbes and destroy them in few minutes. Then antibodies. What is antibodies? Antibodies form the third level of defense. Antibody third level of defense. Here we should know that antibodies are specific substances produced in response to the antigenic material. Antigens are specific substances which, when introduced into the body, give rise to the production of the antibodies. They are produced in the body to help fight against disease causing pathogens. These form a part of adaptive immune system explained later. If the antibody production in the body is slow and the quantity produced is insufficient to destroy the organisms or neutralize their toxin, the organisms overcome the specific resistance of the host. They invade the tissue, start multiplying very rapidly, injure the tissue cells, cause irritation, pain, swelling, redness, and finally lead to inflammation. The swelling and redness are due to dilation of blood vessel of affected part. However, if the antibodies are produced in the large quantity, the pathogen may be killed. And types of immunity. There are two types of immunity. One is natural, one is acquired. Then the natural is divided into the specific, species, and racial. And the three types. Naturals are three types: species, racial, and individual. Acquired are and two types: active and passive. Again, the active are two types: natural, artificial, and passive is also natural, artificial. Then innate immunity. What is innate immunity? The innate immunity is also known as natural, native, or inherited immunity. Inherited immunity. It is called so because immunity we are born with and is the first line of defense. It is non-specific in nature, meaning it protects a person from all the antigens. Innate immunity acts as a barrier that keeps harmful antigen or pathogens from entering the body. So, what is innate immunity? Innate immunity is a barrier to keep the harmful antigen uh, uh, or pathogens from entering the body. It is present since the birth, therefore, it is generally passed on from one generation to the other. A child may possess resistance to certain disease to which mother is immune to. That is why breastfeeding is the very important because child obtains immunity from the mother. Natural immunity is divided into the following three subtypes: specific unit, species unit immunity, racial immunity, individual immunity. What is species immunity? The species immunity means immunity among the member of a particular species. Many diseases which occur in the human being don't occur in the animal. The disease which occur in the man, it is human beings, it don't occur in the animal case or vice versa. For example, disease such as the measles, mumps, polio, syphilis, leprosy, cholera, etc., are found only in the human beings, but it is not shown in case of the animals. This type of immunity is largely due to the difference in the body temperatures, although metabolic, chemical, physiological, and anatomical differences between the different species also play an important role. Then, then racial immunity. What is racial immunity? It means the immunity present among the races of the same species. Some are immune to certain disease while other members are susceptible. For example, in the human beings, Jews are immune to the tuberculosis, but Negroes are more susceptible to it. On the other hand, Negroes have high degree of immunity to malaria, dysentery, yellow fever than white. Complete racial immunity is very rare. Races differ from one another primarily in the degree of immunity. This type of immunity party depends on the some physiological difference that exist between different individual and party depends upon the nutrition, cleanliness, differences in the mode of life and genetics of the members. Individual immunity. It means immunity present among the individual of the same species and same race. It varies with the different individuals of the same species and the same race. The resistance of the same individual may also vary from day to day and time to time. An individual may be very susceptible to one disease organism and very resistant to another. An individual in good health may never catch cold, but if he is weak and exhausted, he could catch cold frequently. Individual resistance of an individual also depends upon the physical state of the individual and varies with the other factors such as diet, fatigue, and exposure. Acquired immunity. The immunity acquired during lifetime of an individual is known as the acquired immunity. It is associated with the presence of specific circulating antibodies. Acquired immunity differs from the innate immunity in the two respects as given below. It is not inherent in the body, it is acquired during lifetime. It is specific for a single type of microorganism. Acquired immunity is of two types, active and passive. Active acquired immunity, if the patient actively participates in making his antibodies, the immunity is called active acquired immunity. There are two methods of acquiring active immunity: natural active immunity It may be acquired in the two ways. Immunity acquired as a result of an attack of disease. An individual may become immune to certain infectious disease by actually contracting them and recovering. It is entirely specific, which means if a person recovers from a smallpox, he is immune only to the smallpox, but this does not prevent him from contracting mm, typhoid or mumps. This immunity of the body is due to the presence of antibodies, which the body develops uh, as a part of the reaction or response to the presence of invading bacteria or the other poisons. In some diseases, the antibody production is so great that immunity may persist for the lifetime of the individual. The disease like typhoid, diphtheria, measles, mumps, smallpox, whooping cough. On the other hand, there are some diseases such as the pneumonia, influenza, dengue, fever, infection where antibody formation is not prolonged and thus there is only temporary resistance. Second and third attack are also possible. Immunity acquired subconsciously on account of the subclinical infection. This type is also called immunity acquired by carriers. It develops gradually after repeated exposure to the small doses of the infecting organisms. Uh, the doses of such pathogens or toxins, although are not sufficient to give rise to disease, they are enough to stimulate the production of the antibodies. Such persons are not suffering from the disease, but they are carrier of the disease. Infectious diseases such as the diphtheria, yellow fever, and typhoid affect children more frequently and more seriously than adults. These adults develop immunity to such disease. So, artificial active immunity. This type of immunity is acquired by immunizing agents, that is antigens such as vaccine or toxoids. It is a method to protect person against infection to which they may be exposed in future. The antibodies formed are similar to those formed by natural infection. It has been found that for an effective immunity against many diseases, two or more injections are required, which are given at intervals of 1 or 2 several weeks. Passive acquired immunity. If the patient does not participate in making antibodies, the immunity given from outside is called passive acquired immunity. Here, the patient is immunized by prepared antibodies, and the body cells don't take any part in the production of the antibodies. There are two methods acquiring passive immunity: natural passive immunity and artificial passive immunity. The immune system. Uh, uh, Immune system is a general term for a complex series of cell factors and processes that provide a specific response to antigens, cells, and organs of some immune system. Primary, the immune system consists of leukocytes, dendritic cell, mm, macrophages, and different lymphoid organs, that is primary secondary lymphoid organs. Coordination among all of them is required for the development of a full immune response. Blood vessels, lymphatic systems connected these organ tissue cells, um, uniting them into a functional whole. A variety of brief idea of types of cells organs is given here. Cells of immune system. Leukocytes. The leukocytes are called the blood corpuscles cell or WBS. Leukocytes are WBS and WBC. WBCs. These are mainly involved in immune response, recognize and neutralizing invaders such as bacteria, virus, and cancerous cells. The leukocytes are of two types. Mm, Glycosides and granulocytes. Granulocytes are characterized by the absence of granules in their cytoplasm and include lymphocytes and monocytes. The lymphocytes further include the B lymphocytes, T lymphocytes, and natural killer cell. Granulocytes are white blood cells that are characterized by the presence of granules in their cytoplasm. They are also polymorphonuclear leukocytes. These includes neutrophils, eosinophils, and basophils. Dendritic cells. Dendritic cells are named for their appearance with the resembly with the dendrites of the nerve cells. And macrophages, monocytes remains in the blood for the 8 hours and then migrate in the tissue when they differentiate and become macrophages. Primary lymphoid organ, bone marrow. All the blood cells include B and T lymphocytes originate from the bone marrow in the human. In the developing embryo, initially they develop from the yolk sac and then by the fetal liver. Thymos, the site of the maturation of the T cell is the thymus. Thymus is situated above the heart and is below bed, which is the further divided into lobules. Lymphatic cells. The lymphatic system consists of a network of the lymph vessels and lymphoid organs, especially the lymph nodes. Secondary lymphoid organs. The secondary lymphoid organs are the organs where the cells and immune system actually fight off pathogens and foreign substances. These are located along the lymph vessel. Lymph nodes are bean shaped and are densely populated with the lymphocytes and dendritic cells. Spleen. The spleen is a large secondary lymphoid organ present on the left side of the abdomen. It plays major role in producing an immune response to the blood-borne antigen. It filters the blood and traps the antigen present in the blood. uh, Mucosal associated lymphoid tissue MALT as the mucosal lining of the respiratory system. Cutaneous associated lymphoid tissue, this related to the skin as the important physical barrier to the external environment and is the largest immune organ. The cells arises in the bone marrow and differentiate into the erythropoietic cell and lymphopoietic cells. The lymphopoietic cell, erythropoietic cells, and later changes into the lymphocytes for the immune system. Other lymphopoietic cells that pass through the fetal liver or bone marrow in human are modified to the B lymphocytes or B cells. B cells have a chemical substance on their surfaces that distinguish from the T lymphocytes. Similar to the T lymphocytes, B lymphocytes move through the circulation to colonies in the lymph nodes and other lymphoid tissue. Both the types of lymphocytes play important role in the immune system. The point of accumulation of these lymphocytes are at the lymph nodes where they encounter all the antigens. The population of the T-pool lymphocytes is smaller than that of the B lymphocytes. Immunity is not the same in all individuals. Certain microorganisms infect one individual while other individual resist the same disease. This means that there are many factors which control this phenomenon. Factors which modify the immune system. The factors are genetic factor, age factor, environmental and nutritional factor, anatomic factor, physiological factor. So the immune system, it depends upon the many factors are depending. A genetic factor, the immune response is under genetic control and varies from one individual to other. Also, there are biological difference in susceptibility to tuberculosis. Age factor, chronologic age influence immunity. These two ages are susceptible to the numerous infections. One group has completely developed immune system, other there is degradation of the many immunologic organs and therefore functions. Environmental and nutritional factors. There is a general increase in the susceptibility to disease under poor living condition. Anatomic factor. This is the first line of defense against the invasion of the microbes and is usually provided by the skin and the mucosal membranes. The type of immune system. The immune system response is classified into two categories: non-specific or innate immune responses and specific or adaptive or acquired immune response. So what is innate immune system? Innate immune system consists of mechanical barrier and phagocytes. The natural killer cell which protect the body from the entry of any infectious agent. These agents, when try to enter the body, are first stopped by the mechanical barriers which form the first line of the defense mechanism. And specific or adaptive immune response, the specific immune response is concerned with the recognition and ultimate removal of the foreign substance or invading infectious agent, is a highly specific sequence. The final outcome of the encounter between the host and a foreign substance is dependent upon the chemical properties of the substance and also upon properties or the type of the host. At this point, it is necessary to introduce and define the term antigen. Antigens are foreign, non-self chemical substances available in organisms that responses by the immune system. The key feature of an antigen is an area of activity is called antigenic determinant. This is located in the surface of antigen molecule and also consists of few amino acid residue or may be an oligosaccharide moiety. Some polysaccharide may also function as antigen, which have a molecular mass over 10,000 daltons. This large size of antigen suggests that they may have a variety of determinants. Smaller molecules may also be made antigenic by combining them with some tissue proteins or polysaccharides. These small molecules are called haptens. Antigens enter the body through a variety of ways, including passage through mucous membrane of respiratory tract and penetrate through the skin by insects, snake bite, injections, and wounds. Once they enter the body, the antigenic nature of the substance is determined by such factors such as chemical nature, dose and size of the antigen, ability of the phagocytes to take up and degrade the antigens for proper infective immune response. How the immune system operates. As soon as the antigen enters the lymphatic system, the immune process begins. The antigen is phagocytized by macrophages, monocytes, or polymernonuclear cells, and major portion of the antigenic material is digested. If the T lymphocytes are stimulated, they will multiply and leave the lymphocyte tissue and travel to the antigenic entry site in the tissue where they become lymphoblast. The latter produce the lymphokines that attract the phagocytes to engulf the antigens. If the B lymphocytes are stimulated, then remain in the lymphocyte tissue and convert the plasma cell to produce antibodies. Cellular immunity, when the T lymphocytes are responsible for the immunity, when the T lymphocytes are responsible for immunity, it is known as cellular immunity. It is also known as cell mediated immunity or tissue immunity. The mechanism by which is immunity develops is given below. The antigens of many fungi, protozoa, selected viruses, bacteria stimulates the T lymphocytes and sensitize them. The sensitized T lymphocytes then enter the circulation and migrate to the site where the antigens are detected. And the antigen site lymphocytes revert to immature cell called the lymphoblast, which produce a series of low molecular weight proteins known as the lymphokinase. The lymphokinases are extremely active and can be classified into different categories. One lymphokine known as the chemotactic factor, migration inhibition factor prevents microphage from moving away, microphage aggregation factor, microphage activating factor. Antibodies, what is antibodies? Antibodies are specific substances produced in the body in response to antigenic matter to fight the disease germ. These serve as the protective agent against the organism and kill them. The antibodies may be found in the fluid part of the circulating blood as well as certain tissue and are mostly produced in the body. Antibody may be univalent or multivalent. Univalent antibody is one in which each molecule of the antibody combines with one molecule of the antigen. A multivalent antibody is one in which each molecule of antibody combines with the two or more molecule of the antigen. Structure of antibodies. And the basic antibody molecules consist of four polypeptide chains, two identical heavy or hydrogen chains, and two identical light or Al chains. These two chains are joined together by sulfur to sulfur linkage to form Y-shaped structure. It is given in the figure 7.3 structure of the antibody molecule. It is a Y-shaped structure, and this heavy chain consists of about 400 amino acids. It consists of amino acids, antibodies, 400 amino acid, while each light chain has the 200 amino acid. Constant and variable regions exist within each polypeptide chain. The amino acid is the constant region of both light and heavy chains are virtually identical among the various antibodies. The amino acids of variable region vary among the hundreds of thousands of the different antibodies. The variable regions of the light and heavy chains combine to form a highly specific three-dimensional structure which gives the specificity for the particular antigen. When the antibody is treated with the papain A proteolytic enzyme, it gets hydrolyzed to give three fragments by getting the cleaved. Types of antibodies. Different type of antibodies have been identified based on the difference in the heavy or chain hydrogen chains. The five classes of immunoglobulins are explained in given below. Immunoglobulin A, this is the first antibody which appears in the circulation after stimulation of the B lymphocytes. It is largest antibody in term of size. Because of size, IgM remains in circulation. The percent of IgM 5 to 10% of the total antibody in the serum. Immunoglobulin G. This is the major immunoglobulin in the normal human serum. Mm, and the immunoglobulin A, this antibody represent about 15 to 20% of the human serum immunoglobulin pool. This antibody exists in the serum. Immunoglobulin E, this antibody IgE plays a major role in the allergic reactions by sensitizing T cells to certain antigens. This class may play a role in the active immunity to parasitic infection and also commonly associated with the immediate hypersensitivity disease such as the asthma and high fever. High fever. It is a monomer. Antigen antibody. When a molecule of the antigen and the antibody are brought together in a solution, antigen antibody brought together in a solution, they cause specific reactions known as the antigen antibody reaction. A number of different antigen antibody reactions have been observed test tubes experiment and the humans as well as in animal. These reactions depend on the numerous factors such as the nature of the antigen, environmental condition, and origin of the antibody, etc. The reactions are very useful in the laboratory diagnosis and various diseases. For the development of specific resistance, antibodies interact with the antigens in such a way that antigen is altered. This alteration may result in death of microorganism that possess the antigen. There are different mode of antigen antibody interactions. The five mechanism of antigen antibody interaction with the type of antibody involved are as follows: neutralizing antibody, antitoxins, agglutinin, precedins, and opsonins. The five mechanism of antigen antibody interactions are neutralizing antibody. These antibody react with the viral capsid and allow hydrolysis of the enzyme. And antitoxins, these antibodies combine specifically with the toxins and neutralize the toxicity side of toxins. Agglutinins, these are the antibodies which react with the antigens on the surface of the organism, such as the bacteria. This action causes a aggregation of the organism and this enhances the phagocytes. Precipitants, these antibodies react with the dissolve with the antigens and convert them to the larger aggregates. Opsonins, these antibodies encourage phagocytes by forming bridge between the parasites and receptor sites of the phagocyte, thus enhance the action of the phagocytes. So, in this summary, we learn how the immune system work, immunity can definition of the immunity system functions, and immune response, and immune system activates helpers, and the antibody antibody. And the last unit is your genetics and molecular biology. And this unit describes the experiments that prove DNA as the genetic material, describes synthesis of DNA by the process of replication, describes synthesis of the RNA by the process of transcription, explains RNA and ribosome in the protein synthesis, describes biosynthetic proteins, and defines the gene expression and explains the regulation of the protein. The chemical basis of heredity. In the molecular biology experiment, DNA as the genetic material and associated with the cellular processes by replication, transcription, and translation. It will be good to keep in mind some of the following facts. In cell, chromosomes bear the genes, and in which hereditary information is stored. Prokaryotes have only one chromosome, whereas the eukaryote have several pair of chromosomes. The number of chromosomes varies and is specific for a particular species. The eukaryotic, which has the several pair of chromosomes, are within the nucleus of each cell. The overall structure and function of the various kinds of cell of a multimolecular organism and in the cell of unicellular organisms are dependent on the information present in the genes and the expression of the genes. Genes are segments of DNA and each chromosome has only one molecule of the our DNA. It is well known that DNA is the carrier of genetic information in all the cellular life forms, including most viruses except the RNA viruses and retroviruses, where this role is performed by RNA. The term gene was introduced to donate an earlier hypothetical molecular species which was responsible for transfer of a particular characteristic from the parent cell to the daughter cell. For example, ability to synthesize a particular protein like insulin. It has been shown that the deoxyribonucleic acid DNA, and in a few number of cases, ribonucleic acid RNA perform the functions attributed to genes. The question which arises prior to this, what qualifies DNA as a genetic material? Why not protein or lipid or some other biomolecules? DNA is the genetic blueprint of life. Genetic blueprint of life. It functions as the molecule that carries the genetic information from one generation to other. डीएनए वर्सेस जो डीएनए कंटेंट ऑफ द यूकैरियोटिक सेल DNA is well suited for the duplication or the replication that is producing or leading to produce the another replica or itself. DNA replication. Before going to the explore the DNA replication, let us explore the basic principle operating behind, which is popularly known as uh, Jar gaps rule. The two basic rule of base The two basic rules of base pairing of the nucleotide pairing are adenine A with thymine T. The purine adenine always pairs with the pyrimidine thymine through two hydrogen bonds. Cytosine C and guanine G. The pyrimidine cytosine always pairs with the purine guanine through three hydrogen bonds. It is shown here in the page 85. DNA is synthesized in the cell from the 4-deoxyribose nucleotide triphosphate, namely deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, and deoxythymidine triphosphate, DTTP. Their structures given below. Structure given deoxyadenosine triphosphate, deoxyguanosine triphosphate, DGTP, 2-deoxycytidine 5-triphosphate, DCTP, and thymidine triphosphate triphosphate, DTTP. The structures are given below. The ribonucleotide are linked to each other with the elimination of a pyrophosphate ion PPI to form 5,3 phosphodiester linkage. The phosphate is the rapidly hydrolyzed to the phosphate ion in the presence of enzyme pyrophosphatase. The base, the nucleotide I, how it converted to the nucleotide 2, then nucleotide 2. Then nucleotide 3. Since this hydrolysis is irreversible, it helps to drive the reaction to completion. The deoxypolynucleotide is built by repetition of these reactions. Uh, the DNA, which to be replicated as a linear array of the bases of the two strands, is given. mm, DNA molecule has one old and one new strands. In actual synthesis, the precursors don't align in a row but are bound to join one by one in a growing polynucleotide chain. Double standard parent DNA, the vertical dotted line represents the hydrogen bonds. Separate stands for the parent DNA. Alignment of the bases of the deoxynucleoside triphosphate precursors along with the two-strand polymerization of the precursor to give the two identical daughter DNA molecule. DNA replication employs a large number of structural proteins and enzymes, each of which plays a critical role during the process. An important enzyme involved in the DNA replication is the DNA polymerase. Three different type of DNA polymerase are known. And three different type of DNA polymerase known. These are referred to as DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. These cat- these catalyze the polynucleotide formation reaction with the nucleotide 1 being substituted by growing polynucleotide chain. DNA polymerases also possess the exonuclease activity, which are important for proofreading or double checking. None of the DMA polymerase can initiate a new polynucleotide chain, that is nucleotide 1 in polynucleotide formation reaction must be in the oligo or polynucleotide chain, which is called a primer. The RNA primer is synthesized by an enzyme called primase, which is a type of RNA polymerase before DNA replication can start. Then there are three types of RNA in the cell, namely the ribosomal rRNA, transfer RNAs, transfer RNA, tRNA, and messenger RNAs, mRNA. So three type of RNAs are there: transfer RNA, ribosomal RNA, messenger RNA. You would recall these have mm, similar chemical structure. That is, these are all polynucleotide in which the individual nucleotides are linked by polydiester linkage. However, they differ with respect to the size, secondary structure, and function. So all the structures are almost it looks same. The RNA synthesized on DNA template by an identical chemical reaction which is similar to that of the polynucleotide formation reaction studied in the previous section. This process is called transcription. The precursor in this case are ribonucleoside triphosphate as against the corresponding deoxyanalogous required for DNA. Further, thiamine is not found in the RNA. Instead, there is uracil, adenosine 5 triphosphate, guanosine 5 triphosphate (GTP), cytidine 5 triphosphate (CTP), uridine 5 triphosphate (UTP). Structure of ribonucleotide triphosphate, the precursor in the RNA synthesis. DNA contains sequence of bases known as the promoter sites. Promoter sites make the beginning of the transcription. And the DNA synthesis where both the strands are replicated. RNA polymerase binds at promoter site on DNA and initiate transcription. Similarly, there are termination signals where RNA polymerase stops and releases the newly synthesized RNA. Transcription is a continuous process in the RNA 5 5-3 direction without any break. The product RNA are very long strands containing thousands of the nucleotides. The DNA template is conserved and neither strand appear in the resultant RNA. Both DNA strands in the template are necessary for accurate transcription. However, the role played by the inactive strands is not clear. Similarity between a DNA replication and RNA transcription. Similarities are each process require corresponding nucleoside triphosphate as precursor. Each case, DNA replication, RNA transcription. In both cases, polynucleotide chain grows from the 5' to the 3' end side. The reaction involves the nucleophilic attack of the 3 OH group of the following chain on the 5' phosphate group of the incoming precursor. Each case, the reaction is driven to completion by hydrolytic removal of the pyrophosphate ion. In each case, the base sequence in the product is determined by specificity of the base pairing with the DNA template. The protein biosynthesis. The process of protein synthesis involves the process of protein synthesis involves the decoding of this code by the process called translation. The pathway of protein synthesis is called translation because the message in the language of the nucleotide sequence on the mRNA is translated into the language of amino acid sequence. This linear flow of information in the form of code from DNA to mRNA and then protein is referred to as the central dogma of molecular biology. The genetic code, uh, DNA RNA contain the information that coded for specific amino acid. However, it was not clear by the process it was coded. The 16 possible combination being AAAT, AG, AC, TA, TT, TG, TC, GA, GT, GG, GCCA. These are the combination 16 possible combination. However, these 16 combinations are still not enough to code for 20 amino acids. A series of three nucleotide bases on a DNA molecule is called triplet. A set of three nucleotide bases on a mRNA molecule is called a codon. A set of three nucleotide bases on the tRNA molecule is called the anticodon. Since 64 codes are possible for 20 amino acids, there are more than one code possible for some of the amino acid characteristics of the genetic code. The genetic code is specific. Specific codon always codes for a single amino acid. It is degenerate. The, and the degeneracy for a number of energy level having the same energy, and the possible codons and amino acids they code are given in this table. You can observe that only two amino acids, tryptophan and methionine, have a single codon, while the other amino acids have at least two codons, and the leucine and the serine have many as six codons as possible. Structure and roles of the ribosomes. This complex of many ribosomes along with a single mRNA are known as the polyribosomes and polysomes. The ribosome synthesis. Each ribosome synthesis one copy of the protein molecule encoded by the mRNA. The endoplasmic reticulum with the ribosome engaged in the protein synthesis is called as the rough endoplasmic reticulum. Activity of the amino acid, and the peptides are less stable than the free amino acid. There are amino acids must be activated by expenditure of energy so that peptide synthesis become thermodynamically possible. Secondly, amino acid have no specificity for the base mRNA codon, therefore they don't recognize them. At this point, it is advised to recall the structure tRNA. This reaction attachment of amino acid require ATP and is referred to as the activation of the amino acid. Formation of the amino acid when it reacts with the ATP, it gives AMP and PPI. The formation of AAtRNA is catalyzed by specific enzyme. It's called the tRNA synthesis. This helps in the recognizing the correct acid almost without any error. The transfer of amino acid from the AAAMP to the specific tRNA, the reaction is driven to complete the hydrolytic removal of the PPI pyrophosphate ion catalyzed by inorganic pyrophosphate. The enzyme tRNA synthesis has active sites for binding to tRNA and corresponding to amino acid. For example, methionine tRNA synthesis is involved in synthesis of the met tRNA. When the amino acid treated with the ATP and the end of tRNA, it gives amino acid tRNA. The sequence of bases in mRNA determines the sequence of amino acid to protein. Mm, In order to complete the initiation stage, the large ribosomal unit binds to the complex and produce the initiation complex. The second tRNA unit, tRNA having the amino acid group, bind to the matching codon site A, that is amino acid binding sites. Elongation of the polypeptide chain. uh, Once the initiation of the tRNA is in the place, all the available tRNA approach and try to attach to the A site. However, the tRNA whose anticodon is complementary codon present in the mRNA at A site is successful. The tRNA with the two amino acids, a dipeptide now become the P site, and this stage become it is holding the growing protein. The ribosome site down three bases and one codon mRNA, exposing a new A site by the action of another enzyme called the translocase. It is important to know here that peptide chain grows stepwise with addition of a single amino acid starting with the amino terminal and ending with the carboxyl terminal. Codons of mRNA are read from 5' to 3' termination polypeptide chain formation. Elongation process of a protein synthesis continues till the ribosome meet any of the codon UAA, UGA, and UAG. These are called as stop or termination codon and are signal of the end of the process of the protein synthesis. There is no tRNA is complementary to the stop codon. Therefore, the process of protein synthesis stops. This enzyme is called the releasing factor binds the empty A site and frees newly made polypeptide chain from that last tRNA. Small and large subunits of the ribosome fall apart and release the mRNA molecule. The mRNA can then be reused for retranslation, or it may be degraded depending upon how much of that particular protein is needed. When the no more protein is required, all mRNA molecules are eventually degraded. The regulation of gene expression. Gene expression is the whole or complete process by which the gene is able to lead to form a functional product like a protein. Essentially, all the cellular process that you have read in previous section constitute the term gene expression. We can say that the sequential flow of information from a DNA to the protein through the two processes is called as the gene expression. A requirement of the various protein in the living organism vary according to its physiological state and environment. Also, some protein must be synthesized in the larger amount than the other. Some protein must still others are synthesized mainly in response to the prevailing environment. The concentration of the protein in the cell depends on the rates of its synthesis and degradation. In the following subsection, we will consider only the regulation of the rate of protein biosynthesis in prokaryotes. Regulation of the protein biosynthesis and prokaryotes. In the control operates mostly at the transcriptional level. A well-known example illustrating the principal synthesis of protein is the induction of the beta glycosidase of E. coli. These bacteria grow very well in media containing glucose as the energy source and carbon source. Under these conditions, the bacteria synthesis very small amounts of beta galactose. If the same bacteria are transferred to the lactose containing medium in the absence of glucose, it starts synthesis beta galactosidase and grow equally well. Lactose is said to be an inducer, and beta galactosidase and inducible enzyme as against other glycolytic enzymes which are synthesized under this condition. The latter are called conjugative enzymes. Introduction of the beta galactosidase is also caused by the beta galactoside Sidase. Most commonly used inducer is isopropyl thiogalactoside. Since it is not hydrolyzed by the enzyme, its concentration remains constant in the growth medium. If the bacteria growing on lactose are harvested, washed, and transferred to a glucose medium, the synthesis of the beta galactosidase is again reduced to the minimal level. At the same time, the cell does not have to synthesize such proteins which are not required. An outline of the mechanism of the switching on and switching of the enzyme synthesis as per requirement is as follows. Several structural genes Genes specifying separate polypeptide chains are found to be continuously arranged on the bacteria chromosome. Collection of these genes and the control elements is referred to as the operon. Example, the lac operon. Uh, the lac operon is responsible for synthesis of the three enzymes, namely beta galactosidase, galactoside, and permase, and thiogalactoside transacetylase. The corresponding three structural genes are continuously located and referred towards ZYA genes respectively. These are immediately preceded by the control elements, namely the operator and the promoter genes. Together, these five genes constitute the lac operon, which is responsible for synthesis of the protein. Uh, and the regulatory I gene produces its corresponding mRNA called I-mRNA, which in combination with the ribosomes and translation causes the synthesis of a specific protein called the repressor. The protein has a high affinity for and binds specially to the O gene. This binding prevents transcription of the lack of operon. The synthesis of the lactose metabolism is prevented as depicted. This situation prevails the absence of an inducer. It binds specially to the repressor to give rise to the inducer repressor complex. The repressor has no affinity of the O gene. In the absence of repressor binding to the operator, the structural gene are mm, transcribed to the lac mRNA and regulating in the synthesis of specific protein. Ready. Regulation regulation of protein biosynthesis eukaryotes. In the eukaryotes, the protein biosynthesis is regulated mostly at the translational level. And changes in the translational pattern in the eukaryotes occur during the cell differentiation. The regulation of protein biosynthesis in eukaryotes is more complicated. As mentioned above, it operates at the transnational level. It involves reversible activation, deactivation of initiation factor, which are required for starting the synthesis of a new polypeptide chain. This regulation is achieved by phosphorylation and dephosphorylation of the initiation factor. Another mechanism which operate mostly at the embryonic stage involves masking of mRNA by associating with some protein or consequent stopping its translation. So in this uh, in this summary, DNA is genetic blueprint of life. We learn it functions as the molecule. DNA is the genetic blueprint of life. It functions as the molecule that carries on the genetic information from one generation to another. So it all the unit is over. Subsequent flow of information from DNA to protein through the two process called the gene expression. This requirement of various proteins in living organisms vary according to its physiological state and environment. This calls the regulation of the gene expression. Prokaryotes control operates mostly at the transcriptional level. Eukaryotes, the operation biosynthesis regulated mostly at the translational level. Okay, it's all over. Okay.